Good morning to everyone out in YouTube land. All right, so we've been covering the mechanism of hormones and cell signaling. Arguably the most important topic that we will cover all semester, because this is how cells communicate, how they are able to know their place in the multicellular universe. And we have begun this sort of grand chart. And just to summarize, but not to write on the board, we've got steroid hormones. And the key point being, can you diffuse across the plasma membrane or are you restricted to the outside surface? So steroid hormones and other nonpolar molecules, they can diffuse across the plasma membrane. Consequently, the receptor for the hormone, which is really what makes the hormone have some kind of action, the receptor is cytoplasmic. And typically it's hanging out in the cytoplasm. When it binds the hormone, also known as the ligand, when it binds the hormone, it now is going to be released and diffused into the nucleus where it acts as a transcription factor. So what we can say then is that steroid hormones act at the level of gene expression, and consequently steroid hormone effects take a while. So if you are given a steroid, you're having asthma or some kind of chronic inflammation, and you take a steroid, you can inhale it, you can get a shot, whatever, and within 15 minutes you start to say, wow, I'm feeling better. You would have felt just as good if you had taken an orange Tic Tac and dissolved it under your tongue. Of course, not the mint Tic Tacs. It has to be orange Tic Tacs. It has to be dissolved under your tongue. Meaning that steroid hormones cannot have an effect in under a couple of hours because all the different kinds of gene expression activities, transcription, RNA processing, probably that RNA, when it gets into the cytoplasm, is going to be put into the Golgi, and it's going to take a while. On the other hand, we have the other class of hormones that are carrying a charge of some kind, typically small amino acid, individual amino acids or amino acid polymers, small proteins, peptide hormones as we call them. These can't diffuse across the plasma membrane. They are going to be interacting with a cell surface receptor. And the cell surface receptor system, we can break that down into two, again, broad categories. The receptor tyrosine kinases, and that is what insulin uses, and actually a variety of different kinds of critical hormones and things involved in cell division, all kinds of stuff, use receptor tyrosine kinases. In that case, the cytoplasmic side of the receptor acts as a kinase, but its substrate is itself, and it cross-phosphorylates, usually in pairs. And so you have that phosphate there, and in this case, that really big negative charge is recruiting a range of other signaling molecules, and we'll look at some in a bit. And these, in turn, are sending the signal downstream. Receptor tyrosine kinases are going to be very fast acting, on the order of 10 or 15 seconds. Then we've got the other broad class of peptide hormone-based systems, and this is known as the G protein coupled receptors, the old GPCRs. And G protein coupled receptors are kind of historically where a lot of the story begins, and it's a bit more of a complicated dive, and so there's a lot going on here. In all these systems, we've got the first message, which is the hormone, the extracellular hormone. In order to do something, it has to generate the second message, which is cytoplasmic. So the way we break down the G protein coupled receptors, I think, is useful by looking at these two enzyme systems that are tucked right inside the membrane that we can generically refer to as the effector. So we've got the G protein acting on its effector. The effector then, as we're about to see, is the final bifurcation spot. There are two effector types. One is adenylyl cyclase, which produces cyclic AMP, and we'll get into the other effector in just a moment. All right, so now, cyclic AMP is a big deal. because, as we were seeing in bacteria, this acts as a sort of stress signal. If the bacteria starts to run low on sugars, cyclic AMP comes up, and that is what allows these different kinds of metabolic pathways like the beta-galactosidase system to turn on. In eukaryotic cells, cyclic AMP does a number of really critical features. Specifically, there is a kinase, and we know kinases add phosphates to their substrate. There's a kinase that is known as protein kinase A. Protein kinase A, abbreviated in the biz as PKA. This is a cyclic AMP-dependent kinase, meaning that it only activates in the presence of cyclic AMP. So, PKA is a cyclic AMP-dependent kinase. And it begins to phosphorylate a whole range of other proteins. And what makes this thing really interesting is the first protein that it phosphorylates is in fact another kinase. And so we have what is known in the biz as a kinase cascade. And it is a rapidly amplifying system. So this is an example. When we are talking about the breakdown of glycogen, which I think we mentioned in our discussion of biopolymers, glycogen is our equivalent to starch. It's not quite as long a chain as starch, so I don't think you can make a great pudding out of it. But glycogen is a glucose polymer stored in the liver. So if you ever do long distance running or exercising and you carbo load, what you're doing is building up more glycogen. I mean, there's a limit to how much glucose you can have in your blood. If it gets above, like 120 milligrams per deciliter, you start pushing into diabetes. So when you're carbo loading, you're not just filling your blood with glucose. What you're doing is filling your liver with glycogen. Glycogen is a glucose store. So when you get that adrenaline bump, one of the things that happens is adrenaline is going to act on the liver cells, and it's going to interact with an enzyme or with a receptor that is coupled to adenylyl cyclase, and it goes like this. We have adenylyl cyclase. It gets tickled by the G protein subunit. It begins creating cyclic AMP. So ATP is becoming cyclized, and we can look at a structure if you're really interested. Cyclic AMP then in turn is going to activate PKA, protein kinase A. Protein kinase A is going to now phosphorylate one of its substrates, and in the liver, in this specific case, we've got the next enzyme is an enzyme known or called phosphorylase kinase. Phosphorylase kinase, in turn, is phosphorylating its substrate, which is glycogen phosphorylase. And in turn, finally, glycogen phosphorylase is able to take glycogen and break it down into glucose. Now, we've drawn this as a linear flow, but in fact, it is a massive explosion because, let's just sort of think about it from this perspective. One adenylyl cyclase, let's just say, can start churning out, 20 cyclic AMPs per second. I'm just throwing the numbers out. Now, each one of those cyclic AMPs is going to interact with its own pKa. So now we went from one hormone, one receptor, one G protein, one effector, to 20 cyclic AMP. 20 cyclic AMPs are activating 20 pKas. Each one of these pKas can phosphorylate its substrate at 20 times per second. And so we now have 20 to the 20th phosphorylase kinases being phosphorylated. Each one of those begins phosphorylating glycogen phosphorylase, and then we've got, within seconds, a whole ton of enzymes that are ready to break down glycogen into glucose, and whoosh, you get this massive glucose flush, literally in 10 to 15 seconds. So the advantage is, for this kind of system, we've got these proteins in the cytoplasm, and they simply are just hanging around. Until they get phosphorylated, they're not active. But once they get phosphorylated, they go to town. And it is this massive amplification that makes this thing work. And so that's why it's known as a kinase cascade. And when we start looking at some figures here, it is not uncommon in different pathways to have a kinase and a kinase kinase and a kinase 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 and a kinase. So we've got all these different kinases phosphorylating other kinases that in turn phosphorylate other kinases that in turn phosphorylate other kinases until finally somebody does something. And this is a way of amplifying a signal very, very quickly. So this is classic kinase amplification system. And at each step, we would be negligent if we didn't throw in, at each step, there are going to be the quieting down enzymes that are also activated. These are different kinds of phosphatases. And isn't that sad? Phosphatases always get the short end of the stick. Everybody wants to talk about kinases. Kinases in black, bold letters, and phosphatases in barely easy to see pink letters. Phosphatases are critical, though, because they are doing the cleanup. They're setting things back up. I often think a little bit about the game Mousetrap. Who knows of the game Mousetrap? A few people. And I want to get like an industrial mousetrap that I can set up here and set up where you drop the ball and then this big Rube Goldberg event happens, as it's known. And we've seen this on TV. So you have this massive effect occurring from this single event. And then finally we'll hit like a dominoes. So at the same time, the phosphatases are the little elves that are setting this thing back up and bringing the little cage back up, resetting the teeter-totter, all the different things. So phosphatases are, in fact, quite critical. So let's not give them let's not shortchange them. But it's the kinases that get all the cool advertising, because they can get you into some trouble. Now, this means that we go from essentially a metabolically quiet cell to a cell that is just going ape in seconds. And that was a fairly significant event. And so the experimental work went that if you take the hormone, glucagon is one of the hormones that does this, and you inject that into the cell, nothing happens. But if you take cyclic AMP and inject that into a cell, look out. Cyclic AMP is potent stuff. Don't be jacking around with cyclic AMP unless you mean business. That is one of the effectors. There is another effector. And that effector is activating kinases as well, but in a slightly different way.
Does the glycogen phosphorylate the glycogen?
Glycogen phosphorylase is actually breaking down glycogen, and it may be removing a phosphate that allows the glucose to diffuse out. And then there's also a glucose phosphorylase that is the sort of the other side of it that is blocking glucose from diffusing and allowing it to be put back into glycogen. Because one of the things that happens a lot with sugars is that you accumulate them against their gradient using some sort of active transport system. And then in order to keep them from diffusing back out, you have to modify them. And so that's kind of a standard thing that you do. But we'll look at a diagram here. And part of this is, I think, the terms, the names, the enzymes came about before people really knew what exactly was going on. But that is what happens. So it's not the best name. I'd rather it be called glycogen turned into glucose ACE, but it's not. All right, so Although in terms of things that I really go, wow, we need to rename it, it's not on the level of ATP synthase. So I'm not willing to write my congressman about this one. You can. All right. The other effector is the following. It is an enzyme. It's actually going to be shaving a little bit of the plasma membrane. So adenylyl cyclase is effector 1. Effector 2 is phospholipase. So this is effector 2. And the immediate question is, all right, what are the second messages that phospholipase is generating? Adenylyl cyclase generates cyclic AMP. Phospholipase, as a lipase, it breaks down, it was identified because it breaks down lipids. It shaves a little bit of the plasma membrane that it's right next to. And it generates two different molecules. So a way to think about this is if we remember our fatty acid coupled to glycerin to make a phospholipid, we have this structure. This is just a bit of review, and it's a way to sort of conceptualize the fundamentals so it's easier for you to remember what's going on. The fatty acid tails are attached to the glycerin backbone, and then we've got the phosphate group and a decorating group like a choline. All right. So phospholipase is going to shave a little bit of a specific phospholipid that's in the membrane right by it. And it generates two different molecules. One is a molecule called diacyl glyceride, which is, of course, abbreviated as DAG. And that's essentially shaving off the phospholipid, or the lipid part of this molecule. And you have the glycerin, and it's got two acetyl groups on it. The other molecule that is generated is diffusible, because this is still actually stuck in the membrane. The other molecule is part of the sugar group that is here, and it is inositol, which is a sugar, inositol triphosphate. So this is a specialized phospholipid that's hanging out right next to the effector. Inositol triphosphate is abbreviated as IP3. So we've got two second messengers now. Diacylglyceride and inositol triphosphate. And they're doing two separate things. And get one on the board here. I'm going to cant you so you can just crane your neck over there. Hi. All right. And here's what they're doing. Diacylglyceride, it's going to activate an enzyme, a kinase known as protein kinase C, PKC. So diacylglyceride is activating protein kinase C. Protein kinase C, like protein kinase A, has a bunch of substrates that can begin phosphorylating. It starts that whole cascade. And inositol triphosphate is actually doing something a little bit different. Inositol, IP3, this is diffusible. And it's going to bind to a membrane vesicle structure that is part of the calcium sequestration system. If you recall back when we were looking at the relative amounts of ions inside the cell or outside the cell, we always were looking at the fact that sodium is always much higher outside the cell than inside, potassium is higher inside than outside, And we made passing note about, look at calcium. Calcium is like at 10 to the minus 5 molar in the cytoplasm. It is sequestered. It is in vanishingly small amounts. And it's actually being held in a variety of sort of vesicle systems and in the different kinds of reticulum. In the muscle cells, we'll see there's the sarcoplasmic reticulum, which holds calcium. And inositol triphosphate is going to bind to this calcium sequestering system, and it triggers a release of calcium. Now we talk about calcium, if I was to say, why do you need calcium? You'd say, to build strong bones. That's why you're drinking milk. You don't need to be drinking milk. You can get calcium from other sources. Regardless, calcium also is a potent trigger of a whole bunch of different kinds of kinase systems. Specifically, one of the generic systems that calcium activates is a protein known as calmodulin. Calmodulin is a calcium-binding protein, and it in turn activates the calmodulin kinase, known as the CAM kinase. So we've got PKC getting turned on, and calmodulin, which acts on a bunch of things and generates CAM kinase or activates CAM kinase as well. So these are ways we can dramatically change the cell's metabolism in just a matter of seconds. Now, a couple of aspects of this. First off, eukaryotic cells, in order to carry out this really fast response, we have to go ahead and invest in protein synthesis, building these different enzymes, knowing that they're not needed now, but they are subject to phosphorylation and activation. And so we take the approach, when stuff happens, we got to be able to move really fast. And so it is worth the metabolic cost of putting these proteins into the cytoplasm even though they're not doing anything. And they may not do anything for a long time until that saber-toothed cat comes. I mean, it is the ultimate kind of, saving for a rainy day. In that, you don't need glycogen phosphorylase now because you're eating and you're getting enough glucose and you don't need to run like heck. But one day you might. And so it is worth it to have these proteins there all the time, quiet. It does bring about some problems, though, because these are very potent systems that if you strike that match, you better be sure you're ready for dealing with the fire. Because this is what we're about to see is one of the mechanisms by which cancer can exploit the system by activating a kinase that can't be shut down. That's a very bad thing. All right, we'll be getting into this as we continue on. Now, let's see. So we've got two different effectors. We've got adenylyl cyclase and phospholipase. Adenylyl cyclase produces cyclic AMP. Phospholipase shaves the phospholipid to give us diacylglyceride and inositol triphosphate. Diacylglyceride activates protein kinase C, PKC. IP3 acts to release calcium, which in turn turns on calmodulin and a whole range of different things. Just as an example, when a egg is fertilized, there is an immediate wave of calcium across the cell that is changing the polarity. It prevents polyspermy. It activates the egg even before the sperm is fused with the nucleus. It knows it's showtime and we're ready to go. So calcium release is a really big deal. We're very keen on maintaining our our handle on calcium. You don't want to let calcium release in your cells willy-nilly. Oh no. All right, so these are real fast responses. As it turns out, we have some systems in play that allow these fast responses to actually act at the level of gene expression. And that means that we can take this fast response and turn it into something that will be very long-lived, much like a steroid hormone. And this was a discovery that came about after this initial stuff. In one kind of idea or model mechanism of the world, you might think, well, we've got steroid hormones for long-lasting effects because we're turning on new genes. We're going to modify the kinds of proteins that a cell has, and that's going to have a very long-lasting effect, potentially. And that's the role of steroid hormones. These guys are acting on things that are already there, acting really fast, and then just as quickly shutting things down. But it turns out that if you keep your finger on the adrenaline button, you actually can affect long-term gene expression in the following way. Specifically for cyclic AMP, we know a really good pathway. There is a protein called the cyclic AMP response element binding protein. And first off, we call this guy CREB. And second, I think we've seen this term, response element. A response element is the term we use when we're talking about an enhancer. And so this came about from a number of different directions in that there are regions in the genome, enhancer sequences, that bind to a transcription factor known as CREB. CREB itself is binding to cyclic AMP. And so much in the way that steroid hormones are binding their receptor, which then becomes a transcription factor, cyclic AMP can bind and interact with kinases and then also turn into a transcription factor. And what this means is we've got now a cyclic AMP dependent transcription factor. And there are a whole set of genes that have this response element, this enhancer in front of them. When you have a cyclic AMP increase, you can activate CREB. CREB in turn goes into the nucleus and begins turning genes on. What this means is that we can have both short-term effects, like 10 seconds, or how about we just call it a short time effect on the order of 10 seconds. And then if we keep that stress signal going, we can then have long-term effects via CREB and its ability to activate specific genes. And in fact, we were talking about how adenylyl cyclase has been implicated in learning and memory. Turns out CREB is as well. And this is why, and you perhaps have experienced this, you can learn things, sort of memorize a list really fast, and then spit it out for the exam and absolutely not be able to do it by lunchtime. I mean, how many of us have memorized how many stupid organic chemistry formula, and then, I had every single I never learned all my amino acids, but I had every single nucleotide memorized for my molecular genetics exam at 10 a.m., and by noon, not there. And perhaps you have experienced this notion of sleeping on it. That is, if you study a little bit before and you get a sleep break, your ability to remember this is now cemented in a lot better. And that's because it takes time to actually get that synaptic neural pathway with adenylyl cyclase activating cyclic AMP to then actually sort of become quasi-permanent by sleeping on it. And this is, in fact, the molecular mechanism by which we can say studying by cramming doesn't work nearly as well for long-term memory as taking your time over a period of days and making sure you sleep on it, because that's why it works. It takes a while for that synaptic pathway to actually get established, because we're actually going to have to change gene expression in those little neurons in order to remember, oh, this is the structure of glucose. So we have some pretty cool things. Another thing that's really quite interesting is another one of the fly researchers, a guy named Tim Tully, who was also working on learning and memory mutants, and he came up with a whole range of learning and memory fly mutations. He actually started a company looking at CREB, in that they have shown in mouse models, and they have not, I don't think, taken this to the people stage, but you actually can generate flash photographic memory by increasing CREB concentrations in the cells. You allow CREB to activate a variety of different genes, and you can essentially give somebody photographic memory for a time, or give mice photographic memory. What mice remember, I don't know. It's actually a pretty interesting potential therapy for dementia, who knows what. But it's actually a really neat trick. If you're interested, look it up. All right. So we've got then the long-term effects from steroid hormones versus the short-term effects of the peptide hormones. The G-protein coupled receptors, particularly, they have this flash amplification. And then after that, if you maintain it long enough, you actually can begin to have long-term effects. Another side effect of this, not all that nice, is that long-term stress actually begins to have deleterious effects on you, because you're constantly maintaining levels of cyclic AMP at a slightly elevated spot, and that begins to affect CREB, and CREB begins to change gene expression, and it leads to a variety of different long-term stress effects. So let's go to the book figures a bit and see what we can see from this. I'm thinking, if I can get to it, and I hope I can get to it, I'm getting swamped with everything else. There's actually a set of questions looking at cell signaling that would be good exam questions, except they're just a hair too nasty. And so I'm going to give them to you as sort of a homeworky thing. And in case you haven't noticed, I'm quite keen on cell signaling. It is a big deal. So we'll look at a couple of figures. If you have your book, feel free to sing along with us. So these are some yeast cells, and they're responding to something, turning into these little schmoo shapes. And then here is a cell phone, where you have the radio signal coming in and sound coming out. That's a fairly lame-ass example, but there you go. At least the word signal is there. So, thank you. Yeah, we need a high mag of that. All right, we've started out on Monday talking about some of the different kinds of hormonal systems, and I didn't get too much into the vocabulary, but we got sort of a classic endocrine system in which you have a hormone-releasing cell in your brain somewhere. It dumps something into the bloodstream, and then that hormone is traveling all the way down to your toes, where you got a target cell that has the receptor. Now, hilarity ensues. One of the points here is, if you ain't got the receptor, that hormone is invisible to you. You don't care. We also can have not endocrine, but what's known as paracrine signaling. And this is where you've got, rather than an endocrine cell up in your brain, you've got cells in the immediate area that are secreting something. And then the cells around it pick that up and begin to change. This actually happens the way of establishing, like, the eye. You have a central cell that says, okay, I'm it, guys. Everybody form up around me. I begin secreting a little bit of a signal, and cells around it begin to respond and go, oh, we're building an eye. Synaptic transmission, we've already talked about that, where we've got the neurotransmitter, and this is a signaling system. It's not the same kind of amplification in the same way. In this case, we have the target cell, either a muscle and a neuromuscular junction, or another neuron in a synaptic junction in the brain, and the neurotransmitter comes out into the synaptic cleft, binds the receptor, and if in the case of activation, you're going to open up a sodium channel and cause that membrane depolarization. Another really cool kind of effect that we don't typically think about it the same way, but it is contact dependent. When you've got a signaling cell and a target cell, and they may actually be both signaling and targets, when they touch, they know, hey, we've touched. Friend, how are you? We can begin establishing cell-cell contacts. When we start getting into cancer, we're going to see this is a critical thing because tumor cells ignore this signal. Tumor cells do not pay attention to social boundaries. They bump up against somebody, and because they don't have functional contact-dependent receptors, they just keep on dividing, and you build a tumor. There's a range of different systems, but just keep this in mind. The fact that a cell can land on a surface, interact with certain molecules on that surface, and say, I know where I am. I can begin crawling, I can settle down, do lots of things. So, these are all different kinds of receptor ligand type interactions.
So you wouldn't call like acetylcholine a hormone?
I mean, it is a neurotransmitter, but technically it is a hormone. Yes. I mean, I'm live on TV. I don't know. Look up on Wikipedia. Yes, I might ask that as an exam question. Is acetylcholine a hormone? Maybe. Thanks. I don't know. It's a neurotransmitter. This reminds me of my 6th grade health class, in which Coach Green, who, rest his soul, was the sweetest man. But Coach Green, he wasn't a physiologist, and people would ask him questions, and he'd be like, "Don't give me that nonsense, Critchlow." Sean Critchlow used to give Coach Green such hell. And now it's being advertised. Hi. God. No, it's a delightful story. I'll tell you later when the TV's off. All right, so here's an example. We got a set of receptor ligand known as the delta notch pathway. You get into developmental biology, you'll go through this pathway. It is a way of taking unspecified epithelial cells and establishing a cluster of cells that are interacting around the central cell. It's a way you can actually end up building an ommatidia in an eye. All right, so here is some examples of signaling molecules. Adrenaline, epinephrine, if you haven't tried it, Sudafed. Everybody tried Sudafed? No. And I'm not saying this. Sudafed, epinephrine is pseudoephedrine. Ephedrine acts like epinephrine. If you want to know what the effects of adrenaline are, you take a Sudafed. And one of the things it does is it is a brilliant bronchial dilator. I mean, you're getting ready to run from that saber-toothed cat. And if you have congestion in your lungs and nose, you pop a Sudafed, and within pretty darn fast, you can feel it. Anybody have real serious allergies and potentially you can go into anaphylactic shock? You might have an EpiPen in which you carry a needle filled with epinephrine and you jab it. And I have not experienced it, but I can guarantee you that it will happen really, really fast.
My friend Josh has idiopathic anaphylaxis, so he'll just randomly go into it and they don't know what, so he always has to have 2 of them on him.
And have you been around when you use one?
Not directly, but I've heard like eyes blown wide open, could barely breathe, all that sort of stuff.
And then,
use 2 of them,
boom, yeah.
And then he's like, oh, hi.
Yeah. Then, see, from here, who has seen the movie Pulp Fiction? Which, I don't know, I have some different issues with the movie. No one's seen Pulp Fiction.
I started it, but I ain't finished it.
Well, there's a scene where Uma Thurman takes an overdose of, I think what she thinks is cocaine, and actually ends up heroin. And they give her a cardiac puncture dose of epinephrine, and she immediately sits up and flips John Travolta out. All right, I didn't say that. All right, so let's see. Now, cortisol, this is a steroid. So it's going to affect lots of tissues, big, broad spectrum. And how many of you guys ever watch House? It used to be a thing.
Yeah,
right?
I love House.
Well, one of the first things House does is put them on steroids. Not to be mean, but when your doctor says, well, let's go ahead and try a course of steroids, what he is essentially saying is, I don't have a freaking clue what's going on with you, but what the hell, we'll try this and see what happens. That's what saying we're going to put you on a course of steroids means. It's doctor speak for hell if I know, but we'll see what this does. Steroids work in a very broad way and affect lots of different cells. So then we've got some sex hormones like estradiol, a steroid derivative, cholesterol, secondary female sexual characteristics, insulin, protein, glucose uptake, testosterone. I guess, are these in alphabetical order? Yeah. Thyroid hormone, derivative of the amino acid tyrosine, stimulating metabolism on a lot of different cells. All right, and now more signal molecules. These are different kinds of peptide hormones. Many of these you've heard. Things like epidermal growth factor, platelet-derived growth factor, nerve growth factor, histamine, causes blood vessels to dilate, and generally leads to inflammation, and so you could take an antihistamine. Nitric oxide is one that we don't go into too much, but nitric oxide is a gas, and it is a smooth muscle relaxant. It's probably still actually prescribed that people that begin to have the beginnings of heart disease, you might struggle with something that is known as angina, in which you've got arteries that lead to the heart are becoming blocked. And so you probably are getting close to having a stent, but not yet. And as you put the heart under stress, the blood flow is not able to keep up with the demand that the heart has for oxygen, and you begin to feel chest pain. And what you can do is pop a nitric oxide tablet. One of the things you could do is take a small nitroglycerin tab, dissolve it under your tongue. Nitroglycerin decomposes and generates nitric oxide, and that causes the smooth muscles that line the arteries to relax, and you have increased blood flow. Not that I would know about this or ever condone this. If you've ever heard of something known as poppers, that you can sniff and your face flushes really, really red. Good. Don't start. Don't look it up. It is a nitric oxide derivative, and that breaks down to nitric oxide. Whoosh! It causes a dramatic blood vessel opening, and gives you a bright red face and flush. Viagra is also using nitric oxide, and it actually causes some very specific effects. And if you don't know what those effects are, you can look it up. Next, neurotransmitters. So yeah, we're going to throw neurotransmitters into signaling molecules. We got acetylcholine, GABA. GABA is a big inhibitory one. Acetylcholine is a big excitatory one. A lot of nerve muscle synapses, and also in the central nervous system. Nicotine is having a very interesting effect that if you don't smoke, don't start. But if you want to try again some hormonal experiences, take a little dose of nicotine in the form of nicotine chewing gum. And initially, it'll do some unpleasant effects, make you feel like you're going to throw up. Not really fun. But over time, you begin to develop the addiction to it, and you actually remodel your synaptic response to it to where it has all kinds of interesting relaxation effects. All right, contact-dependent signal molecules. There's actually a lot of them, too. And anything good here? Yeah, salivary glands. Acetylcholine. I don't know about acetylcholine. All right, so what we're showing here is the complexity by which hormone receptors and hormone interaction can have some pretty dramatic changes on what a cell is going to do. So in this sort of stylized graphic, if we've got hormone and hormone receptor ABC, when that hormone is binding to its receptor, the cell is happy, it's maintaining its survival. This actually is a critical part doing tissue culture. You've got to make sure you have all those different hormones present. Then you add D and E, and now instead of just surviving, you begin to divide. On the other hand, if you add F and G, you begin to differentiate. You go from just being a fibroblast to forming some kind of nerve cell. You don't have any of the hormones, and you end up dying. Apoptosis is actually a cell-specific death, cell suicide, as it's called. It is not the same as just leaving, cells out on the table to dry up and rot. It is, in fact, an active effect that the cells are saying it is time for us to blow up, to take ourselves out of the picture. And the initiation of apoptosis causes DNA to be broken down in a very specific way. And it is essentially a very specific shutdown of a cell. It's a term that maybe we can talk about later. So, all right. This is, again, another good figure. Yeah, we're going to knock off here in just a moment. Oh, what time is it? All right. You're right. We should knock off. This is a good figure, though, that if you're down with this, you're starting to get the big picture here. We got the fast response. Then we got the slow response. The slow response is going to be acting at the level of gene expression. And we'll pick up and discuss a few more ins and outs of this on Friday. Join me, won't you? The university disavows anything I've said.